I'd like to introduce our next speaker, Dr. Isabella Grumbach from University of Iowa. The title of her talk is Calmodulin Kinase as Nodal Integrator of Calcium and ROS-Dependent Signaling in Blood Vessels.
Thank you very much for inviting a practicing cardiologist / large blood vessel cardiologist or blood vascular biologist to this meeting. Now, to make matters worse from your perspective, I received an email about a week ago which said, could you please also talk about redox signaling in cardiac myocytes? So instead of trying to take you through redox signaling in vascular cells and cardiac myocytes, I decided to concentrate on a particular aspect, and I will try to integrate the findings and highlight the opportunities and challenges of the field. Vascular disease, heart failure, arrhythmias, cancer, and asthma. What do all these diseases have in common? I'm sure you've heard this many times. Excessive oxidative stress. And this concept has been around for long enough so that there are now pills, berries, soft drinks, even facial creams with antioxidants which promise to keep you healthy and make you look better, and they lead to a perfect world. Now, the bad news that I have for you is that antioxidant therapies in this global generalized fashion have in clinical trials mainly failed. And that is probably because concise redox-responsive disease pathways have remained elusive. Redox signaling. What is redox signaling? Well, it just means that particular signaling pathways can be activated or shut off by electron transfer events such as oxidation or reduction. Now, if you find this too theoretical, I think a way how many of us think about this, there's a disease-initiating event, hypertension, hyperglycemia, which leads to increased ROS production, and then to ROS-sensing, disease-inducing signaling mechanisms leading to vascular disease. But to me, this is more like a big black box. And the reasons for this are somewhat elusive, but some aspects of why antioxidant therapies have failed are that ROS isn't produced everywhere in the cell and at all times. It's really a localized event, and it's a temporarily localized event as well. So ROS are mainly produced in caveolae lipid rafts, in endosomes, in mitochondria. And since many reactive oxygen species have a very short half-life, it's at least interesting to speculate that their main effect will be on the neighboring signaling pathways and structural proteins. So what certainly does not happen, as is depicted in this cute and slightly fuzzy slide, is that reactive oxygen species shown in red are just swimming everywhere in the cytosol and they're eaten up by antioxidants. So, one way to look at redox signaling is to also step out of the limitations of just looking at one pathway and another pathway, which is what many signaling people like to do, but to think about how to integrate signaling pathways and see how they intersect and may, in combination, lead to disease-initiating events. And as you all know, calcium is one of the most important secondary messengers in the cell, and there are numerous ways to see how calcium signaling and ROS signaling may interact. For example, there is good evidence that numerous calcium channels, for example, the L-type calcium channel, may be regulated by increased ROS production. On the other hand, NADPH oxidases can be, for example, NOx5 directly activated in a calcium-dependent fashion. NADPH oxidase subunit P47, for example, is phosphorylated through PKC, which is calcium-dependent. Calcium is taken up into the sarcoplasmic reticulum through SERCA, and there are numerous papers at least suggesting that ROS-dependent events modulate SERCA function. The ryanodine receptor and the IP3 receptor are responsible for releasing calcium for the sarcoplasmic reticulum. And again, there are numerous papers suggesting that superoxide and hydrogen peroxide affect their activity. Lastly, mitochondria are a significant source of intercellular ROS production, and they're also one of the greatest calcium stores. So there are numerous opportunities for calcium and ROS signaling to interact. And there are a number of fairly well-defined effectors in this regard. PKC signaling certainly is a science in itself. Protein tyrosine phosphatases and ChemK2 have emerged as effectors which may integrate calcium and ROS signaling. ChemK2, or the calcium and calmodulin-dependent kinase 2, is actually strongly expressed in numerous cell types, and which also includes the vasculature. And I apologize for including a slide of the aorta. So this is ChemK2 expression, and as you can tell, there's robust expression in smooth muscle cells, in the endothelium, is probably best shown here, and also in cells in the adventitia. Now, what makes ChemK2 interesting in this particular context is that it can be activated through calcium, leading to autophosphorylation at threonine 287, and based on data which is fairly old by now and very, very well established, ChemK2 for the longest time has been thought to be a decoder of intracellular calcium events. Now, it's been known for a number of years now that in addition to activation through autophosphorylation, ChemK2 can be oxidized at 2-methionine residues in the autoregulatory domain, leading to sustained increased activity. In smooth muscle cells, which is the main focus of my lab, we and others have shown that ChemK2 signaling affects calcium homeostasis and has some impact on smooth muscle contraction and blood pressure, hypertrophy through calcium-dependent signaling pathways, inflammation, migration, proliferation, and apoptosis. And the vascular biologists in the audience will, of course, think of migration and apoptosis as ROS-dependent events, and many of the findings on this slide were really made before appreciating the function of chem kinase in redox signaling. Since I was asked to also speak about cardiac myocytes, here one slide on ChemK2 function in cardiac myocytes. There are numerous publications, more than a slide could fill, which have described seminal findings about chem kinase in cardiac myocytes, which include hypertrophy, similarly to calcium-dependent signaling events, electric remodeling through chem kinase phosphorylation of, for example, the L-type calcium channel of the ryanodine receptor, and events surrounding inflammation and apoptosis. And there is now equally good data over the last 10 years that chem kinase is activated through oxidation of these 2-methionine residues in disease states or under treatment conditions which are known to increase intercellular ROS production, for example, angiotensin infusion. Now, we started out using an antibody which was given to us by a long-term collaborator, Dr. Anderson, who's now at Johns Hopkins, and this antibody fairly reliably detects oxidized chem kinase, but does not detect non-oxidized chem kinase. So we were, for example, able to identify oxidized chem kinase in atherosclerotic plaques, but really didn't see much evidence for this in blood vessels of so-called normal patients. In diabetic retinopathy, we detect chem kinase in the endothelium of capillaries in the inner plexiform layer. However, the oxidized form was only present in patients with diabetes. Similarly good is the data on OXChemK2 as a correlate of high ROS disease states in humans in myocardium. Here's an example, the recent publication on atrial fibrillation. So this is human tissue from the right atrial appendage showing significantly more oxidized chem kinase in patients with atrial fibrillation versus sinus rhythm. And the overall expression of chem kinase was not different between the two groups. On the right, an example of a heart failure model. So this is a Western blot, again using the same antibody, showing higher levels of oxidized chem kinase in diabetic patients after MI versus non-diabetic patients. Now, an antibody is only as good as it is, and not surprisingly, for at least one project, we were asked by the reviewers to prove that there is that oxidation of this site occurs in vivo, at least in a high ROS disease state. And so we embarked on a journey of mass spec experiments where we first made a number of peptides which correspond to the autoregulatory domain of chem kinase, and we made different peptides which are oxidized and non-oxidized, and we were actually very happy to find out that an elution pattern which corresponds to the oxidized form of peptides containing this oxidation site were only present in a high ROS disease model, but not seen at baseline. So one of the difficult questions in redox signaling is, does this modification mean anything? So is inhibiting oxidative chem kinase activation sufficient to prevent vascular pathology? In order to show this, we used a mouse model in which these two oxidation sites at methionine 280, 182 were mutated to valines. And I'm going to refer to this model as the M2V or MMVV model. And indeed, we were able to show in different scenarios that just point mutating these two sites is sufficient to affect vascular pathology. So this is a very simple experiments done by a student in the lab who isolated smooth muscle cells and performed apoptosis assay in response to hydrogen peroxide in migration assays, and she found that the MM to VV mutation significantly decreased the propensity of the cells to become apoptotic or to migrate. We used this system in vivo in the large blood vessel biologists' model of arteriogenesis. So this is a model in which very simply the left carotid artery is ligated and the right carotid artery undergoes eutrophic outward remodeling. And while this was definitely present in wild-type mice, in mice which were lacking chem kinase delta, as well as in our chem kinase MM2VV model lacking the oxidative phosphorylation sites, this did not occur. So there is equally convincing data from myocardium. So this is an atrial fibrillation model which is driven by infusion of angiotensin by mini pump, and when the mice undergo an atrial pacing algorithm, and as you can see here, wild-type mice which were angiotensin infused did develop features which remind of human atrial fibrillation about 60% of the time, whereas the angiotensin-infused mice with this MM2VV mutation did not. Similarly to mice lacking NADPH oxidase subunit P47. So this story led to a concept in which angiotensin induces ROS production, which activates chem kinase for oxidation, and oxidized chem kinase then phosphorylates the ryanodine receptor, leading to calcium leakage and a higher propensity to develop atrial fibrillation. So lastly, this is unfortunately very busy, and I'm just going to highlight two particular mouse strains. So these are mice which were made diabetic by streptozotocin injection and then underwent ligation of the left anterior descendant artery to induce a form of diabetic cardiomyopathy. And the survival of the wild-type mice was actually pretty dismal. However, this was significantly improved in the mouse model with the MM2VV mutation shown in blue, as well as other mouse models in which chem kinase was lacking. So one area of surprise, and which has been actually reproduced by other groups as well, is that this is actually not a one-way street. So we didn't only find that ROS activated chem kinase, we also found evidence in different models, both in vivo and in vitro, that that chem kinase also regulates ROS production. So just very simple examples. So these are again the smooth muscle cells from these MM to VV mice versus wild types. And in response to a cytokine mix, they produce significantly less ROS than the wild type mice. So the lessons that my lab and our collaborators have learned from these exercises is that it is possible to identify single redox signaling pathways, and it's really key to develop relevant tools like an antibody which reliably detects the modification you're interested in, and also probably important to have an in vivo model which mimics the redox modification. And very often you need an integrated approach to demonstrate the physiological relevance of the modification you're studying. There are numerous gaps. Some are related to this project, and we are still, are the fodder for discussions at weekly lab meetings. But from the big picture perspective, I think that it is still very difficult to understand different aspects of redox modification. These are both technical gaps as well as conceptual gaps. The technical gaps is we don't really have a good technology to resolve intracellular ROS spatially or temporarily, and it's still very difficult to predict which amino acids will be modified and will indeed impact protein function. Conceptual problems include it is really unknown whether there are cascades of ROS-dependent signaling similar to kinase phosphorylation events, and also most of the focus in redox signaling has been paid to oxidative modification of amino acids. However, fatty acids certainly also undergo modification, and this will probably lead to a far more complex picture than what we are seeing right now.
Thank you, Dr. Grumbach.
Jose Lopez from Seattle. So the fact that you can detect these oxidized CAM-K2 in tissues in disease states indicates that it's not really regulatory, but it seems to be irreversible. So why is it not reversed by methionine sulfoxide reductase?
It is.
It is.
It is. Yes. So we've done some studies showing that methionine sulfoxide reductase A at least appears to reverse oxidative activation of chem kinase.
And so is that actually the defect in these disease states, that the MSR is defective?
So that's a very good question. So we have a number of experiments in MSRA knockout mice, and they are not the opposite of the chem kinase model. So the answer is it's probably more difficult than this.